It's not a word, Wayne.
It is a word. I looked in the book.
No, I looked in the book as well. It's not in the Scrabble book.
Well, it's in the chemistry book. It says key definition, then defines it. How can you have a definition if it's not a word?
You can't do that. I can't look in a geography book and say, oh, that's a place, I'll put it in my Scrabble word, because you're not allowed places in Scrabble.
Oh, but this isn't a place. It's a word, like any other word. It's got a definition. You don't define Paris.
If you had a chemistry Scrabble, fine, but it's not a chemistry Scrabble yet.
Okay. When we were talking about Scrabble, we were playing Strategio.
Dunno, but it was a long word.
That was a long word. So, we're on to amino acids.
We are.
Let's first go through the general formula in case you don't do biology.
Yeah, so an amino acid is a thing. It forms proteins, you know from that from GCSE. And it's got two parts to it.
Yep, it's got the carboxylic acid part on one side, and an amine part on the other side. And you'll notice that this central carbon only has 3 bonds at the minute.
So it needs another one that can be formed with, well, a variety of things.
And this is why there's lots of amino acids. They all have these things, but this changes so you can have a variety.
You can. As all residual groups, it can be any length carbon chain or an OH or anything, but there is a simple one.
Yep, it's just a H, and this is called glycine.
It is.
There's 20 amino acids in the human body.
And they're all used to make, they're all used to make proteins, which you need for, you know, stuff.
Like hair.
Nails.
But mostly stuff.
Mostly stuff.
So this is glycine, it's the simplest one. We're not going to go through the others because you don't need to know them off by heart.
No, but you need to know that there are others and appreciate that.
Yeah.
There's a very swish thing about amino acids in knowing.
There is indeed. They can interact with themselves.
Which is questionable at best. And they can form something called a zwitterine, which is a very long word.
It is.
And that's where the H plus, my H going to side, switches. So it sort of forms an acid and a base at the same time. So this will essentially
positive overall.
disappear.
Yep.
So that'll be gone here.
That's gone to here. It's actually attached there. That's positive overall because it's gained a proton.
That'd be negative overall. You could write the negative there, or you can write it here, but you can't write it there.
You could write the positive there. And notice it's not taking that oxygen, so you may be penalised depending on how strict the particular exam is.
Yeah.
So just don't.
Just don't do that. So this is a zwitterine. But when does that happen? When?
Well, it actually happens at particular pH values.
Okay.
So this pH value is called the isoelectric point, which is another fun word.
So isoelectric point is lovely. For glycine, it's 6.5, and it's different for each one, isn't it?
It is indeed. So it's not the same if you change this R group. It has different isoelectric points, so different pHs at which it can do this Zwitter ion, which this can exist.
So if this was 6.5, it would form this thing where it has no overall charge.
Yeah. But what happens if you change the overall pH? What's it going to do?
Well, that's a good question, Wayne. So if we change it to something, make it neutral.
Okay, so perfectly 7.
Yeah, we put it as 7.
This pH is greater than the isoelectric point.
It is. So what it's going to do is try and get back to that point.
So we can imagine this being this pH of 7 because it's more base-like than this. Even though it's neutral, it's still more of a base than this.
It's going to try and add an acid to itself to get rid of that.
And an acid is a proton donor, so to neutralise this, it's going to donate a proton.
So it's going to act as an acid. So it's greater than an isoelectric point. So that's greater than iso. So it's going to act as an acid.
Yeah, it's essentially, it's neutralising. So if this, even though this is neutral, it's more basic, so it's got to be an acid to neutralise it.
Yeah.
Same thing, if you had a pH of less than 6.5.
It then acts as an alkali to reverse that change.
So it would attach it back here. So it would have the OH group again.
Yeah.
Now, when it loses this proton, this goes back to its original charge and that side stays negative. When it gains this, that's going to go and this side would stay positive. So that's something to remember.
So it'll form a different ion. And that's really handy.
Yeah, and that will form. You don't have to know the isoelectric points. It would be given to you in the question.
You just need to know that they are all different for different things.
Yeah, and you'd have to know this pH difference of what it's going to have to act as to neutralise it.
Yeah.
Think about where that's going to attach or lose from. And that's a big word we've learnt about there.
That is. I quite like it.
Strategio takes a couple of hours. I've only done 1 minute, so we should carry on with that.
We should do that. Okay.